all. I extend an amiable and fervent welcome on behalf of Modern Institute of Pharmaceutical Sciences Indore, who has assembled this AICTE-sponsored on-site international conference with the motive National Products and Drug Development Towards a Healthier Life in 21st Century Research, Innovation and Idea. I, Purvat Himani, truly regard it is an honor and privilege for standing here as your moderator. Nothing succeeds like success. No one else could be better incarnated by these, but our diligent session chair, Professor Dr. Kamlesh Dashura, HOD Institute of Pharmacy, Vikram University, Ujjain. Sir has more than 70 international and national publications to his credit. We are honored to have you, sir. It does not matter how slowly you go, as long as you do not stop. I hereby welcome our eminent speaker, Professor Dr. Malikarjuna Pichika, who is felicitous for the radiant words. He is the Associate Dean, Drug Delivery, International Medical University, Kuala Lumpur, Malaysia. His field of excellence being pharmacology and clinical research, he has 53 publications to his credit. Sir will be articulating on the topic National Products in Drug Development, Recent Advances, Applications, Challenges and Opportunities. Over to you, sir.
Hi, is my voice audible?
Yes, sir, it is audible.
Okay, could you please allow me to share my video because
Yes, sir. Sure.
I cannot share my screen, so I think you need to allow me to share my screen.
Sir, share kar. Sir, please share your screen.
Yeah. Okay, just to confirm, is my
Yes, sir, your PPT is visible.
okay? Is my screen visible, madam?
Yes, sir.
Hello.
Yes, sir. Sir, please start.
Okay, ma'am. I hope my voice is clear.
Yes, sir. Is it audible? It is audible.
May I know that how much time do I have? But actually, I have another appointment at 12:30. So
Sir, you have
I'm not sure whether
Yes, sir. You
I will stop at 12:30, madam. Is it okay?
Yes, sir.
Okay, thank you. Maybe I can deliver only half an hour. Okay. Hi. First of all, good morning to everyone. Okay. Thanks for inviting me. Of course, I can understand there will be lots of technical glitches. Okay. That I cannot deliver my full lecture, but I see whatever the best that I can do. Okay. First of all, this is my disclaimer. Okay. Whatever I am presenting here, it's only my views. It is based on my expertise, understanding, and experience. Okay. And there is no way these views are from my institution. They do not represent. Okay. And most of the information that is obtained here, that I put up here, is obtained from the reliable web sources. Okay. And the relevant references are cited at the end of the presentation. Okay. The audience, just this is my sincere request. Okay. If you would like to adopt any of those things, please confirm and double check before you would like to adopt any of these concepts into your research. Okay. Thank you with this disclaimer. Now the question is, does natural products still play a role in drug development? because nowadays the people are talking about synthetic chemistry is greater than the natural products. Okay, and computational-related drug discovery is going to make some wonders. Nowadays, the people are dreaming about artificial intelligence. Okay, so we are going to see that whether natural products still play a role in the present era. Okay, that's what we are going to discuss. Okay, I'm going to share my views. And of course, those who are very familiar with natural products, okay, they should be, by this time, they should be familiar. Natural products doesn't mean that it's only a plant material, okay? It could be an animal, it could be a microbe, it could be even human, okay? And of course, this slide shows a representation that natural product could be anything, okay? It should be a living organism. Of course, even soil is also considered as a natural product because it contains some microbes, and that microbes will produce lots of secondary metabolites. Of course, this is one of the famous animals. Okay, then 40 different types of medicines are obtained from this one, and recently you might have heard that the people are trying to do a heart transplantation using a pig's heart in human. And one successful operation was recently conducted in US. Okay? That shows that how promising this animal is. Okay? And according to the FDA, new drug approvals, okay, it is classified like this. Okay, it could be a biological macromolecule, unaltered natural product, a botanical drug. That means botanical drug means it's a plant material, a mixture of plants, or a mixture of semi-synthetic product plus plant material. Okay, it could be a derivative of the natural product, which we call it as a semi-synthetic, because the starting material is coming from the natural product. Or it could be a completely synthetic product, okay, usually which is obtained from the random screening or modification of the existing drugs. Okay, and of course in synthetic drugs, sometimes the people try to synthesize drugs using pharmacophoric features that are present in natural products. Okay, and that is represented by S star. Okay, of course vaccines, and also some kind of a natural product mimics. Okay, mimics. That is, when we look at the structure, they look very similar to the natural products. They could be from completely synthetic, or they could be from a synthetic drug which contains a natural product pharmacophore. And here I am presenting the new drug approvals since 1981 until 2019. Okay? So in total, there are about 1818 products are there. Now, you may, as I told you that I'm presenting the new drug approvals since 1981 until 2019. Okay, the reason for that is why I have chosen 1981 is that's where actually a lot of advances were happened in genomics, okay, in proteomics, lipidomics, metabolomics, a lot of omics, okay, the computer-aided drug discovery techniques, and the artificial intelligence. Okay, and many people assume that because of these advances, the people assume that the drug discovery process will be fast. We can find a better drugs. Okay, that is the assumption that the people have. But actually, we will see what was the reality and what actually the statistics is telling to us. Okay. A total of 1,881 drugs were approved by US FDA since 1981 until September 2019. Okay. And here you can see that in total there are 463 drugs, okay, that are completely from the natural sources, that are completely synthetic route. Okay. whereas the biologicals are 346, okay, and things are 142, okay, and except that all others are, okay, somewhat related to the natural resources. Actually, this diagram shows, okay, a cumulative percentage of the products from the different sources. Actually, from this you can see that, okay, still the natural products, even in this era of omics, in the era of artificial intelligence, in the era of computer-aided drug discovery, still it is dominating. Okay, so that is the take-home message. Okay, still about 60% of the drugs are still coming from natural product. It's not directly from the natural product. It could be got the heat from the natural product, then making some synthetic modifications or creating a structures very similar to the natural products. So all together, okay, including botanicals. Now you can see that NB stands for natural botanicals. Nowadays, the FDA has started approving the drugs, okay, which are plant powders or mixture of plant powders. Now this shows the source of the drugs, okay, over the years, okay. Actually, you can see that because most of the people will concentrate on synthesis. Okay, you look at the synthesis bar, which is represented in yellow color. Over the years, it is gradually decreasing. Okay, over the years, it is gradually decreasing. When you look at the natural products, okay, and the natural botanicals and natural semi-synthetic derivatives, you can see that it is almost remaining constant throughout. Okay, of course you can see little bit fluctuations here and there. But since 2010, actually you can see that more and more biologicals are getting approvals. Okay, but people thought that all the biologicals are going to over this one. Through product and synthetic, in my opinion, it is highly unlikely, because the reason for that is there is lots of quality control issues, storage issues, okay, and that's why I think still synthetic and natural products still they have their prominence in coming years. And if you look at this one, okay, these are the small molecules, okay, and if you look at it, there is a spike in the between 1985 until 1989, okay, then afterwards it is almost remaining constant, okay. And here when you look at the biologicals, you see that this is a 2015 and 2012 onwards, you can see that there is an increase in the FDA approvals of biologicals, okay. And in general, we will see that on an average, in the case of biologicals, 5 to 10 drugs per annum, okay, approvals, but in the recent years, it has gone up to 15 to 25. Whereas here, the average approvals, okay, per year is between 20 to 30. Okay, small molecules, except there is a high in between 1985 to until 1989. If you look at the newly approved small molecules, okay, since we do not have much time, I will concentrate on this part, which is very important. Still, you can see that natural products are the drugs which have origins from the natural sources still has not dominating. And here also you can see that among the newly approved small molecules, okay, you can see that still even in these years also, okay, 2016, 2017, 2018 also, you can see that natural products are still contributing to about 10%. Okay, yeah. And when you look at the synthesis, it is about 15 to 18%. Okay. And new drugs approvals from the natural sources. Suppose if you look at the new drug approvals only from the natural sources, that means It could be a pure natural product, it could be a botanical natural product, or it could be a derived product, or it could be a synthetic product which contains a nitrogen, sorry, natural product as a pharmacophore. Okay, you can see this. Okay, direct means the compounds which are directly isolated from plant sources or from the natural sources, and indirect means it could be a natural product mimetic, or it could be a synthetic product which contains a natural product pharmacophore or natural product analogs. Okay, and it is about 50-50%. Okay? Of course, this is the distribution over. Okay? Then, if you look at the disease indications for how many diseases actually you can see that the natural products are useful. Okay, just we will compare only the natural product derived, okay, that is semi-synthetic, okay, and synthetic ones. Okay, synthetic molecules are there for about 464 disease conditions, whereas natural product derived products are indicated for 356. That means it is suggesting that there are some diseases, okay, which are being treated only by synthetic drugs. That means natural products did not show any effect in some classes. Okay, a few of the examples are antihistamines, diuretics, and hypnotics. Okay, and this is the three classes of drugs where until now there is no single drug that is of natural source is approved by FDA. Then if you look at this top 20 therapeutic categories, basing upon the number of drugs that is approved by FDA, anti-cancer, antiviral, antibacterial, anti-hypertensive, anti-diabetic, okay. But if you look at the top 5 classes of therapeutic classes for which the FDA has given approval is anti-cancer, anti-diabetic, anti-infective, anti-inflammatory, and anti-hypertensive. These are the 5 classes of drugs okay, for which the FDA has given a maximum number of FDA drug approvals. Okay, when I say here anti-infectives means it includes antibacterial, antifungal, antiviral, and anti-parasitic. Okay, so this is the percentage, okay, of the number of drugs that are approved, and here you can see that in each class, you see that more than 60% of the drugs are still coming from the natural products, even in this era. Okay, except for the for these 3 classes, that is anti-cancer, anti-diabetic, and anti-infectives, more than 50%, or almost 60% of the drugs are still coming from the natural origin or somewhat related to the natural. Okay, whereas the number of drugs, okay, in the case of anti-inflammatory and anti-hypertensive, mostly it is from synthetic origin. So all these slides or this information gives some kind of a confidence that still the natural products play a role. Okay, yes, the synthetic chemistry is helpful, computer-aided drug discovery technique is helpful, AI is helpful, but however, they cannot override the prominence of the natural products. That's why maybe even after 1980s or 1990s, if you see that pharma industry, when I say the pharma industry, I'm talking about the big giant companies, okay, like a Pfizer, AstraZeneca, Takeda, those things, okay, and they are not concentrating much on the natural products, okay. They are trying to, they are concentrating more on actually synthetic molecules. Maybe in the recent past, if you have seen, many pharma companies have merged together because they could not able to survive. Maybe one of the reasons could be because they concentrated too much on synthetic products, or that they thought that synthetic is going to be the way. Okay, that's why they ignored the natural products. Okay, and that's why a lot of mergers were happening all over the world. But if you look at the companies, even in India, okay, the companies who relied on natural products, still they could able to exist, still they are existing, still they are doing that. Okay, but the companies who put lots of their R&D investment in synthetic drugs, okay, and some, if not majority, went for mergers. Okay, and that shows. Okay, and the new anti-infective drug approvals. Okay, if you look at this antivirals, mostly it is coming from the natural sources. Okay, antibacterial and also antibacterial. New anti-cancer drug approvals still got linked to the natural resources are still dominating. Hi, my voice is audible or are there any issues? Because I got a message internet connection is low.
Yes, sir, it is audible.
Can I proceed?
Yes, sir, sure, sir.
Thank you. And new drug approvals for diabetes and multiple sclerosis. Okay, and here also in these two, because anti-diabetic one of the common diseases, and the entire world is suffering with. Okay, and the people are expecting that by 2050, almost 90% of the population should be diabetic. If we do not pay special attention to the living style and also the food habits. Okay. The another one is the multiple sclerosis. Okay, and you can see that over the years, okay, the average human life has increased. Okay, from about 50 is now it is about 80 to 85. Of course, in Western countries it is an average human life is about 90 years. Okay, and when the people become old, what happens means they come up with some kind of neurological diseases. Okay, where the brain may not function most actively. Okay, and that's why the people are expecting that, because I think by 2050 also, I think by 2050 there is some calculation saying that the population above 50, okay, in the entire world is more than 60 to 70%. Okay, that means the population size of younger generation will becoming smaller and smaller and smaller. Of course, in that way, I think we should be happy. In India, still there is a higher proportion of young generation compared to other countries. Because there are many reasons why it is so. Now, basing upon the sales, okay, annual sales in US, okay, these four drugs, Lipitor, Humira, Nexium, and Adalimumab, okay, these four drugs are considered blockbuster drugs. Okay, this is based on the information in 2019. Okay, it may not be applicable now, okay, because of the COVID-19. Everything has changed. Of course, now the vaccines, and if you look at the Pfizer's one, okay, and on average, one estimate saying that per second they are earning about $2,000 okay, per second. Okay. And the expected revenue from vaccines to the Pfizer is expected to be around $960 billion by 2025. Okay. So this one, if you see the atorvastatin, among these two, okay, this Lipitor, all we know that it is for cholesterol lowering reason. This atorvastatin, it is came from the fungi, aspergillus, and this one, Adalimumab, anti-asthmatic. It is the combination of the fluticasone and the salmeterol. Okay, this fluticasone is also a natural product mimetic. Of course, it's a human's sex hormones, male sex hormone mimetic. Okay, and salmeterol is natural product mimetic of, again, is the humans. It is a phenylephrine. Okay, or norepinephrine. Okay. So, now these are some of the examples of the natural product drugs which are very famous. Okay, lovastatin, quercetin, artemisinin, and nicafamine. Okay, and for this one, the inventor, okay, the person who invented that is Toyota from China. Okay, sorry, Toyoyo. Okay, sorry, Toyoyo from China received a Nobel Prize for her work on this artemisinin, okay, in 2015. And actually that story is very interesting. I think once upon a time the people talked about Madam Curie, okay, who has consumed this radioactive material, okay, to see the effects. And in the recent years, this is Dr. Toyoyo, is the one actually who took this artemisinin on her own, okay, because of the various reasons, okay, because this is from China, so the people might have some suspicions, okay. That's why she took the artemisinin on her own and has proven that it is an anti-malarial drug. Even now, it is the best anti-malarial drug. If you look at the budget of the US, okay, there is I'm not getting the name of that organization. Even now, they spent about 2 billion ringgits on artemisinin plants, okay, artemisia plants, to see whether they can make a better compounds from this artemisinin, okay. So let us say the natural products as drugs, and what is the major differences between the medicinal chemists and the synthetic chemists. Okay. First thing is natural products is a diverse scaffolds and a structural complexity. Of course, if you look at the structures, even humans cannot even imagine those kind of structures that is existing in the nature. And also they have a high molecular mass. Okay, whenever we talk about a medicinal chemist, the people talk about the compounds of smaller molecular weight or compounds of molecular weight less than 500. This is what they talk. Maybe that is one of the reasons why nowadays the people are not able to innovate blockbuster drugs. Okay, because of this mindset. Okay, they are putting them on 500, 500, 500, and I will talk about it later. Okay, and a large number of sp3 carbon atoms and oxygen atoms. Okay, that means here in natural products, most of the time you will find single bonds. Okay, whereas in the synthetic molecules, mostly we look at the aromatic rings, we look at the double bonds. Okay, that is one of the major differences. And also we will find lots of oxygen atoms. Okay. In synthetic molecules, we will see a lot of nitrogen. Of course, few there will be halogen atoms. Mostly the people use chlorine because it is a bioassociate for many things. That's why, and it's also a small atom. Okay. And higher number of hydrogen bond acceptors and donors. Okay. Whereas in medicinal chemistry, we try to restrict the number of hydrogen bond donors to less than 5 and hydrogen bond acceptors to less than 10. But here in this case, it is 5. And they are very, very hydrophilic. Okay, that's why the people think that they do not have absorption issues. They have absorption issues. Okay? And some they have a greater molecular rigidity. Okay, which I will talk about later. And a Lipinski. Okay, and I will explain on that in the next slide, next few slides. Okay, these are the, if you look at the molecules, it's very difficult to imagine such a molecules exist in nature. And if you look at these big black comb rings, okay, or big rings, that's why they are very, very rigid. Okay, and rigid molecules are required, okay, to disrupt the interaction between the two proteins. Okay? And nowadays, the protein-protein disruptors is one of the investigated area. Okay? Until now, the people thought that it is very difficult to disrupt the protein-protein interactions because the binding space or binding cavity is quite large, and the people have this kind of a mindset, the number of molecular weight should be less than 500. So these small molecules could not able to disrupt this protein-protein interaction. Okay. And these are the famous rules that they use in drug discovery. Okay. And this is one of the things, Lipinski rule of 5, okay, which is widely used. But one thing the people forget. Yes, Lipinski was a scientist at the time working in a Pfizer. Okay, And the Pfizer company has asked him to do the investigation of why the success rate is going down. Then when he investigated at that point of time, okay, I think it's about 20 years back, okay, he analyzed all the FDA approved drugs, and from that he come up with this kind of a criteria. Molecular weight should be less than 500, log P less than 5, hydrogen bond is less than 5, and hydrogen bond acceptors is less than 10. Okay, he come up with this idea because he thought that 95% of the molecules at that point of time, what I'm talking about is about 20 years back, okay, 95% of the drug molecules that are approved by FDA, okay, has followed this criteria. Then that's why he proposed that if anyone wants to synthesize a new molecule, new drug-like molecule, maybe he postulated that the people should consider this. But when the people started considering it, I didn't see any much kind of improvement in the success rates in the drug discovery. Okay. And these are the various rules. Okay, the AICTE is company, okay, a pharma company. Okay, they have their own rules. They call it as a rule of 3. Okay, this is called for a fragments. Okay. Now maybe you people might know that the people what they look at is they look at big molecules. They will chop into small pieces, which we call them as a fragments, and they will put them together in like a jigsaw puzzle, and to see whether it works or not. Okay. Whereas if you see this one 3 by 75 rule, actually this rule is nowadays is being done by Pfizer. Okay. Please remember, once upon a time, Pichika is also the employee of the Pfizer. Now the Pfizer has come up with this kind of a rule, but this is for in vitro toxicity. Okay? And because majority of the drugs are being failed in clinical trials only for 2 reasons. One is the lack of efficacy, the other one is lack of safety. Okay? So this is what they do for toxicity. Okay? And this is the bioavailability score. Of course, nowadays the people use rat, okay, and from the rat they will calculate the percent bioavailability, okay, or fraction of the bioavailability. From that, they will extrapolate to how much is being absorbed in humans, okay. And this is total polar surface area. Of course, from the total polar surface area also, if it is less than equal to 75 angstrom units, then the absorbance will be higher. This is the beyond Lipinski rule of 5. Of course, I do not have much time. Of course, here there is a link. Now, if you look at these drugs that are approved by FDA, okay, if you look at their molecular weight range, then you can see that the drugs that are approved after 1998, you can see that they are much higher than, okay, the molecules whose molecular weight is less than 100. So you can see that there is a change in the trend. Okay, and it could be because of advances in the omics. Maybe it is also could be because of approval of the biologicals. Okay. So, but here the take-home message is, in the drug discovery, still the drug development, still we should consider the Lipinski rule of 5. And these are the very famous examples. You look at this molecular weight, very huge molecules, and you look at these hydrogen bond acceptors. Almost all they have more than 10, and this molecular weight is more than 700. And all these are obtained from the natural products, and until now there is no replacement for these antibiotics. And no one could be able to find better drugs. That is the best way to do that. Okay, from synthetic routes, or from computational methods, or from using artificial intelligence techniques. Okay, these are the antivirals. Okay, of course, the molecular weight is also greater than 700. Here, the interesting point here, if you see this one, a molecular weight is very high, but if you look at this hydrogen bond donors and hydrogen bond acceptors, still they are very less. It's very difficult for a medicinal chemist to design a molecule, okay, having this kind of a molecular weight with less number of hydrogen bond donors and hydrogen bond acceptors is very, very difficult. It's very, very difficult. And also, if you can look at these molecules, you can see a lot of C single bond C. In a medicinal chemistry, what we say that whenever we see such a single bond C, first of all, it is very difficult to synthesize. Okay, people can't imagine that they can synthesize these kind of molecules. Okay, if at all they synthesize, also the cost will be much higher. And they also talk about the flexibility. And since they have flexibility, that's why the people expect that they are not stable. Okay, and of course, these are some of the examples. Then we saw actually the plant products. Mainly they are for anti-cancer and anti-infective. Okay, why is it so? Those who have a little bit knowledge in pharmacology, one of the best science.
Hello, sorry for the interruption, sir. Your session time is over. We have further more sessions in. Can you please sum up your presentation?
Oh, okay. Then I will stop, madam. Thank you.
Thank you so much, sir. What a pertinent session it was. I will now request our students to get clarified with any kinds of doubt and questions.
Good afternoon, sir. Good afternoon, sir. So, sir, what are the challenges we face while developing a new drug? Am I audible?
I'm not sure whether you are asking any questions, but the voice is not clear, so I cannot hear.
Sir, the question is, what are the challenges we face while developing a new drug?
Okay. Maybe if you look at this slide will summarize. This is the main problem with actually the problems with the natural products. I think I will take about 2 minutes and I will stop here. Okay. The problem one is not possible to culture the organisms out of the natural habitat. This is applicable both for plants, even for animals, even for the microbes. Okay. That is one of the challenges. Okay. The plant may grow in India, but the same plant may not grow in US. Okay. But even if the people want to bring this plant to the US, then you can see that the number of the natural products that are produced will be different. Okay, that is one of the biggest challenges. Okay, and what the solution is nowadays is because of lot of omics data. Okay, and in the past the people have tried with the tissue culture techniques for plants, but it didn't. It was not so successful. But nowadays the people are moving towards the microbes, okay, for the drug discovery, because the microbes can grow faster, okay, can replicate faster. So that's why the yields of the natural products are much higher, okay. So these two problems can be overcome. I'm saying from the perspective of the microbes, but still from the plant sources, still there are some challenges, okay. Then the another problem is, sometimes what happens means we take the extract, okay, we do some activity, it is found to be excellent, then we go for isolation and isolate, and you will come up with the same compound that is already reported. That is another challenge that the people face. Okay? So nowadays the people are coming with a de-replication techniques using NMR and mass spectroscopic techniques. That means once you have an extract, you run the NMR first, and you see whether are there any new compounds which are not reported earlier. If there are new compounds, then only go ahead, otherwise just stop, don't work on the extract anymore. That's why if you look at the papers, almost all, on an average I can see that per day, definitely I can see that a minimum of 20 to 25 papers in what is called Science Finder, okay, or in Scopus, okay. about what is it called flavonoids. So it's a waste of time. You cannot get anywhere. Okay. Then the another problem is the natural product. Definitely it may have bioactivity, but since it is having a permeability issues, okay, that's why when you culture, it may not penetrate through the cell wall. That's why it will not show the activity. That's why if you are choosing the anti-cancer activity, your drug molecule is very good, but only the natural product is very good, but it may not penetrate through the cell wall. That's why it will not show the activity. There is another problem. Okay, and sometimes the hormones that are present in natural products is very, very small, and sometimes it's very difficult to quantify. Okay, and that's why there is a lot of techniques nowadays came. Okay, advances in NMR spectroscopy with 600 megahertz, 1000 megahertz. Once upon a time, we need to have a minimum 30 mg of the sample to run the NMR, but nowadays 1 mg sample is sufficient. Okay, so those challenges can be overcome. Okay, and identifying the molecular targets for the natural products is one of the daunting tasks. This one is very, but nowadays there is a lot of advances in metabolics, omics, and if we can make use of that, then we can overcome these challenges. I think I can say that this is the summary of the problems and the possible solutions.
Okay, thank you so much, sir. I will now plead Professor Dr. Kamlesh Dashora, sir, to apprise our students with his words, taking the session towards its conclusion.
First of all, I thanks to Professor Dr. Malikarjuna, Pichika, Associate Dean, Research and Consultancy. Dr. Malikarjuna, you have really given a nice presentation on natural products in drug discovery development, recent advances, applications, challenges, and opportunities. Professor covered the role of natural products, around 40 medicines, new drug approval classifications of around total 1881 drugs. Then also categorize the source of new drugs, that is small molecules or biologicals, then approval process, disease according to disease indications like anti-cancer, anti-diabetic, anti-infectives, multiple sclerosis, and also discuss about protein disruptors and rules in drug discovery. Professor, you have given really a nice presentation. It was an informative and it creates a new dimension for the development of further natural products in drug discovery and development. And I am also congratulate Dr. Anil Khariyaji and Sapna Malvi, who will invite me as a session chair in international conference on natural products for drug discovery and development towards healthcare life in the 20th century, research, innovation and idea. Thank you very much for inviting me, and also thank you to Dipen sir, Nirmal Dongre sir, Nilesh Jain, and Satyen Srivastava and Sapna Malvi. Thank you.
Thank you, sir, for your captivating words. I appreciate our students for being such an encouraging and perceptive audience. We are obliged to have Professor Dr. Kamlesh Dashora and Professor Dr. Malikarjuna Rao Pichika here with us. Hope you all have vigorous day ahead. I will now request Ms. Sanjana Naidu ma'am to chaperone our guests to the auditorium.
Sir, you have comfortable to sit here, otherwise you come with me.
Can I leave or what?
Yes, sir. You can leave, sir.
Thank you.
Thank you, sir.